So now we'll talk about lipids. And I guess this thing is recording, huh? I suppose I want to double check. The heck is the thing? Yeah, it looks like it's recording. So again, there's stuff that's already up there, and this will be, it looks like I'm going section by section to make, you know, the Khan Academy, they try to make them a little smaller so that they're digestible. So it looks like this is just lipids. All right, questions before we start? All right, so lipids are, the definition of a lipid is a nonpolar molecule of living things, or I guess that's the functional definition for this class. There may be a more precise definition out there, but so that includes fats, oils, waxes, steroids, etc. And what it doesn't specify is a particular shape for these molecules. These molecules can be very different shapes. As long as they're nonpolar, we call them lipids. So they're generally high energy molecules because a lot of them are hydrocarbons. And hydrocarbons, the carbon-hydrogen bond that we see in these things is high in energy. It can be burned. It's combustible. Lipids are important because they're for energy storage, right? I'm storing more and more these days, as I ceaselessly remind you. It forms cell membranes are made out of lipids. Hormones like testosterone and estrogen, as we learned about earlier, are lipids, and they are hormones. And so here are some various kinds of, and I don't know why the spinach is included in this picture, but it is. Okay, so hydrocarbons are nonpolar, so are lipids. Hydrocarbons are things like gasoline, so it doesn't mix with water, right? Nonpolar things don't mix with water because water is polar. And if you guys remember the information from Chapter 2, Part 1, you remember that there's oxygen and hydrogen and two hydrogens in H2O, yeah? And the oxygen hogs the electrons, gets a slightly negative charge. The two hydrogens get a slightly positive charge. So the negative region of the oxygen and the positive regions of the two hydrogens make water polar. In other words, having poles, having regions of positivity and negativity. Hydrocarbons are nonpolar. In other words, they're not plus and minus. They're zero all over. Does that make sense? So the fact that they're zero all over means that they don't interact well with things that are polar, right? So we see little droplets of oil not mixing with water as they're poured into water. So triglycerides, that is regular fat molecules, are a primary lipid structure. This is ripped from another textbook, I think because it shows in pretty good detail the structure of a lipid. We have in yellow here 3 lines. These lines are hydrocarbons, CH2 a bunch of times. So 2 covalent bonds to each carbon plus 2 to hydrogens on the sides. That's where you get the CH2. And then these CH2s can be varying in length. The first one is shorter, the second one is longer. And then they can be bent. This one has a double bond where that bend is occurring. So that is what's called an unsaturated fatty acid. Each of these hydrocarbon chains is called a fatty acid. The green thing on the left is called a glycerol molecule. And basically, those are, glycerol is strictly speaking a 3-carbon carbohydrate, right? If you remember carbohydrates having OH groups all over them, there's 3 carbons and each one has 1 OH group. And the OH group, along with the carboxyl group of this, in other words, carboxylic acid group of this fatty acid, the OH here, the H here, by dehydration synthesis, the same process by which carbohydrates are assembled and disassembled are the same processes by which lipids or fatty acids are linked onto glycerols to form a fat molecule for these fundamental lipids, yeah? Is that clear? All right, so we do that 2 more times and we have a triglyceride, which is a standard molecule of what we call fat. And if all of them have single bonds and none of them have double bonds, then it's a saturated fat, like you might get in butter or animal fat. And if there's double bonds in it, then it's unsaturated, like olive oil. And if it's a lot of double bonds, then it's polyunsaturated, like corn oil or vegetable oil. Is that clear? All right. So that's the difference between monounsaturated and polyunsaturated. So the length and saturation of fatty acid chains makes fats different from each other. Furthermore, we can see that this bend that occurs affects the way, if you have many of these molecules, it affects the way the molecules stack one on top of another. If you imagine a big box and you're throwing a bunch of triglycerides into it, if the triglycerides are bent, they're not going to fit into the box as easily. And as a result, ones that are saturated tend to be solids like butter, and ones that are unsaturated tend to be liquids like olive oil or vegetable oil. And the more unsaturated a fat tends to be, the more fluid and less gooey, less viscous it tends to be. Is that clear? All right. So cis versus trans, this is something that's much more relevant for nutrition than it is for microbiology class, but I'll give it a brief measurement, right? If both of the hydrogens in a double bond, remember a double bond shows a place where it is unsaturated. In other words, there's room for 2 more hydrogens. But the fact that there's no hydrogens on the left side of this cis fatty acid means that the ones on the right side of the cis fatty acid can take up a little more space. Molecules like to have a little bit of space. They like to spread out. And as a result, the whole molecule bends. We have a bent molecule on the left, and the hydrogens stay on one side, and that's what causes the bend. The trans form, one hydrogen is on one side, the other's on the other side. And because of that, each hydrogen on the opposite side takes up about the same amount of space, and the molecule doesn't bend as much. Well, as a result, trans fats, even though they're unsaturated, tend to be solid, right? Like Crisco is a hydrogenated oil, and the reason for that is because the molecules are straighter, yeah? And tend to be more solid instead of the bent ones that don't stack well and tend to be more liquid. In any case, they are isomers, which is worse for you. Trans fats are worse, and the primary reason for that is because cis fatty acids are found in biological systems and in nature. If we look in most fatty acids in nature, most of them are cis fatty acids. So your body has very few enzymes that are designed to break down trans fats. So once they're in your system, tend to be a lot harder to get rid of. But again, that's nutrition stuff. That's not so much microbiology stuff. But it's interesting. Okay, much more broadly relevant is phospholipids. If we take our triglyceride and take off one of the fatty acids and substitute onto it a phosphate functional group. Remember the functional groups? One of them was phosphate. And if you can see here, there's a minus sign on this PO4 phosphate. So is this polar or nonpolar? Polar, right? If it has a minus or a plus, it's polar. That means charged, basically, right? So this is polar. The tails, though, are lipids. They're nonpolar. So is this molecule polar or nonpolar? Answer, it's both. It's all of the above. The head is polar. The tails are nonpolar. And that's important. So that makes the molecule what's called amphipathic. That means having polar and nonpolar regions. And things that are that are both polar and nonpolar, do they mix with water or do they mix with oil? Answer, they can mix with both. So the things that are phospholipids are one thing that mixes with both polar and nonpolar substances. Another thing is soaps, right? Soaps, if you look at your shampoo or something, it'll say sodium dodecyl sulfate or sodium lauryl sulfate. That is, the lauryl means a 12-carbon fatty acid, and the sodium is a charged sodium ion at the end. So you have something that's both mixes with oils and mixes with waters. Where's there oil? Where there's oil in your scalp, you're trying to wash it out. You use a soap to get the oils in your scalp to mix with the water in the shower. That's what your shampoo is, a soap. It does that. Does that make sense? Okay. Good to move on? Questions? Okay, so steroids and cholesterol are both lipids. Cholesterol is the precursor to most of your steroids, and you can see the molecular similarity between cholesterol and testosterone and estrogen. Yeah? Which of them is the least polar? Well, cholesterol is the least polar, right? These ones have several functional groups with OHs on them, right? OHs can do hydrogen bonds with water. These have a few. This has only one. Does that make sense? So here on cholesterol, the little OH group at the lower left is the only place that water can touch it. Testosterone and estrogen can flow in your bloodstream because they have a couple of OH groups on them that help them mix with water a little better. Okay. So other kinds of lipids. So these things are called mycolic acids. You don't need to know a ton about these. You should know the name, though. These form the cell walls of Mycobacterium tuberculosis. Have you heard of this guy? Maybe you haven't heard of the microbe itself, but hopefully you've heard the disease it causes, yeah? Very important disease, and a very important disease of poverty. All right, so these fatty acid chains, right, these zigzags is just a really, really long hydrocarbon molecule, yeah? So these are way the heck longer than most phospholipids. Here's the polar part at the very right-hand end of these things, but most of the rest of it is non-polar, and it's, again, way longer, and these are really waxy. They're solid, and they're waxy, and they're really hard for your body to get at, right? One of the things that makes tuberculosis such a nasty microbe is these mycolic acids in their cell wall that makes them so hard for your immune system and your enzymes and so on to attack. And along with antibiotics have a difficult time attacking them. Okay. So isoprenoids you also don't really need to know details about. They are lipids. They are beta-carotene. This is the very top little thing here is the basic unit, and they're modular. We can take a bunch of these and link them together, and you might, using some imagination, imagine how a bunch of these little guys on the top could be linked together to make beta-carotene on the bottom. Yeah? Does anyone see how the thing on the top, you get 1, 2, 3, 4, 5, 6, 7 or so of these, and you can make of these, and you can make beta-carotene. If you don't sweat it. This is not vital information. This is sort of a extra put in there. This molecule is harder to imagine that it comes from isoprenoids, but this is artemisinin. It's a drug that is good against malaria. Okay, so in salad dressings, oil quickly separates from vinegar because oils are non-polar is the correct answer. 3. Good. All right. So proteins. I'm going to stop here because proteins is a rather large enchilada. So I'm gonna stop